We presented this whole exome sequencing of smoldering multiple myeloma this year at ASH. So the study is about 100 patients for which we did the whole exome sequencing and targeted sequencing of one part of the cohort. And so the goal was to define the mutational landscape of smoldering multiple myeloma and to eventually identify markers of progression in a subgroup of patients. So we had defined the mutational landscape with the main mutated pathway, which are MAP kinase and NF-kappa B, as well as the cell cycle pathway and the DNA repair pathway. We also identified a group of patients, like above 50% of the patients without mutations and very quiet genome. So it's one of the hallmark, I guess, of the smoldering multiple myeloma. And so what was interesting is that when we classify the patients based on their clinical stage, the myoclinic clinical stage, we could find mutations in all the stage. So they were not clustered. However, when we separated the patients between progressors and not progressors, we could clearly identify some specific mutation only in the progressor group. So it led us to develop a kind of marker where we actually identify the MAP kinase mutations and the MYC alterations that occur much more in the progressor group, and it does increase the risk of progression and shorten the time to progression in these patients.
When it comes to the clinical utility of this, of how easy it is to detect and then act on those sequences?
Yeah, so these are maybe preliminary data, and I think we need to have more cohorts. But the real question behind that is about early interventions. And we know now that there is a potential interest of treating these patients early on and to try to cure the disease early on and before they develop overt multiple myeloma and they become symptomatic. So the real question here is that we identify these two markers, the MAP kinase pathway mutations and the MYC translocations. So do we have to treat, for example, these patients? We cannot answer yet to this question, but it's definitely something that the community will have to study in large cohort of patients and really find the risk of progression and the markers for these patients and potentially change our attitude and decision in terms of therapeutic.
Is there anything else you can think to mention? Any other details?
I mean, we are looking forward to continue this project and to be able to study large cohort of patients and potentially have a real clinical impact of this study.